So yeah, today we're gonna talk the lung-brain connection and the ventilation brain injury. A little bit about me. So I'm a neurosurgeon who worked 10 years, and I joined the Dr. Reynolds lab because I'm very interested to study the communication between the brain and the lungs. So the agenda for today, first, I'm gonna talk about the types of this is a hot topic today, especially because a lot of patients, they have to use mechanical ventilation. So we're gonna briefly talk about this. Then the background about the brain-lungs connection, a review of previous preclinical research, and the pathophysiology of ventilation-associated brain injury, and some results for my last manuscript. Okay, so first, I'm gonna talk about the types of ventilatory support. So when a patient needs support because problems to breathe, the first type that we have the facial mask, which is a mask that supports oxygen. And in this case, the patient is able to breathe by their own. They don't need pressure. They don't need anything, just more oxygen. The second type, we have a non-invasive mechanical ventilation, which means these patients, they have a mask, but right now there's a machine that delivers this oxygen to the mouth and the nose, positive pressure. And the last one, we have an invasive mechanical ventilation, which means that in this case, the doctors, the physicians, they have to place a tube through the throat, and this tube is connected to the machine, and this machine will take over, or not in some case, the breathing cycle, which means that the machine will deliver air using positive pressure. And this is this stream of ventilation has been used since the 1970s, but right now it is in vogue because a lot of patients, they need mechanical ventilation, and there's a desperate need for devices to help these patients to breathe when they have COVID. And a lot of reports start to appear that when these patients are under mechanical ventilation and they have COVID, they're having cognitive impairment. They're having delirium, they have long-term cognitive impairment even after a hospital discharge. So the first thing that we have to understand is there a connection between the lungs and the brain? So I'm gonna talk briefly about this. So when we are breathing, there's a nerve called vagus nerve. This nerve connects the lung and the brainstem. So will reach the brainstem in a nucleus called nucleo detractus solitarius. This nucleus will communicate with another nucleus called locus coeruleus, and the locus coeruleus will communicate with the hippocampus. So, but this is true. So some clinical studies, they have studied this. So during neurosurgery for epilepsy, neurosurgeons, they do surgery in awake patients, which means that the patients are awake. So when we stimulate, we are able to see that there's a stop in the breathing cycle. So we induce apnea. Also, when the patient have tumors or epilepsy in the hippocampus, this leads to apnea as well. And in the preclinical studies, this was also verified by more electrical stimulation of the hippocampus to stopping in the breathing cycle. Not even that. When we use that machine, the mechanical ventilation machine, in a rodent model, but we apply an enormous amount of air inside the lungs, we are able to observe that we induce hippocampus apoptosis. And apoptosis means cellular death. So applying a lot of volume into the lungs, we are able to kill or to see that cells in the hippocampus die. And finally, in 2019, one research showed that the tidal volume, which means that the volume that we apply into the lungs using mechanical ventilation, it is directly correlated with the hippocampus activity, meaning that more volume means more activity. So the bottom line here is the hippocampus impacts the brain and vice versa. So there's a connection between lungs and the brain, specifically the hippocampus. So there are two pathways that people have been describing since 2013 that might be able to induce brain injury due to mechanical ventilation. The first one is the neural pathway. So remember the nerve that we talk about, the vagus nerve. So this research, they show that when they cut the vagus nerve, even though they apply a huge amount of air volume inside the lungs, they're able to prevent hippocampus apoptosis. And this research, they show that maybe the vagus nerve is responsible for this connection. And if we manage to treat this nerve or do something with this nerve, we are able to mitigate the brain injury or the injury associated with mechanical ventilation. Pathway, it is called inflammatory pathway. In this case, either proteins or inflammatory cells traveling from the lungs to the brain will reach the hippocampus, triggering neuroinflammation and cellular death. So these two research, in order to show that, they assess, using a rodent model, they assess cognition, and they have three groups, the true control group, the other group that was ventilated for 1 hour, and the third group that was ventilated for 6 hours. After 6 hours, they extubate the subjects, which means that they are breathing by their own. They're not under mechanical ventilation anymore. They assess cognition 3 days after extubation, showing that the group that receives 6 hours of mechanical ventilation, they have worse cognitive scores in comparison with the subjects that receive 1 hour or the true control. And not that, the group that had cognitive scores, they also had greater levels of neuroinflammation and cellular apoptosis. To challenge this, the inflammatory pathway, this research called CAMOVE, this research designed a study that had two groups. One, both groups are mechanical ventilated for 12 hours using a porcine model. In one group, this research induced inflammation, lung inflammation, and showing that there's no difference in neuroinflammation and cellular apoptosis after 12 hours of mechanical ventilation, saying that, okay, mechanical ventilation itself is triggering this injury that we're observing. So regardless the pathway, mechanical ventilation is a common factor that is associated with brain injury. Also, all the preclinical studies that I mentioned use injurious mechanical ventilation, which means that the mechanical ventilation settings that these they're not mechanical ventilation. They're not the settings that we are using in ICUs for our patients. So our group thought, okay, how about we test maybe the lung protective mechanical ventilation settings, it is not injuring the brain. So in order to do that, we designed a study that was published this year in March 2021 at Scientific Reports that is called Brain Injury After 50 Hours of Lung Protective Mechanical Ventilation in a Preclinical Model. And the first outcome was hippocampus apoptosis, and the secondary outcome was hippocampus neuroinflammation and biomarkers for brain injury. So the study design was using a porcine model in a mock ICU, comparing the never ventilate group with mechanical ventilate group for 50 hours, using the gold standard in mechanical ventilation. So we analyzed neuroinflammation and cellular death. As neuroinflammation, we use reactive and microglia, and to analyze cellular death, we use apoptosis as a marker. And why we choose like 50 hours? Because a lot of clinical studies, they have demonstrated more than 2 days of mechanical ventilation, it is associated with 10 times greater cognitive impairment in humans. All the statistical analysis that we use are non-parametric, which means that we use, in this case, Mann-Whitney U test, and all the results are expressed as median and interquartile range. And our study was the first long-term preclinical studies on the effect of lung protective mechanical ventilation on the brain on the planet. So we are very proud of it. So the first result that I'm gonna share was cellular death. In order to mark cellular death, we use a TUNELI. TUNEL assay, it is a late apoptotic marker, and clinical studies, greater hippocampus correlate with cognitive impairment. So on the left, we can see a slide from a never ventilated subject, and every brown cell means TUNEL positive cells. And on the center, we can observe a mechanical ventilated slide. And on the left, we can observe a dot plot showing data from the never ventilate and mechanical ventilate group. So there's a considerable difference between the group, and we observe just under 1% of TUNEL positive cells in the mechanical ventilate group, and 31%, just above 31% for the mechanical ventilate group. So think about it. We got young, healthy pigs, human-sized pigs. We ventilate for 50 hours using the gold standard ventilation, and after able to observe that hippocampal cells are dying. The next marker that we used was to analyze neuroinflammation was reactive astrocytes. In order to mark these astrocytes, we use GFAP. Why we're using GFAP? Because it's a neuroinflammatory marker, and also it is easy to add into translational studies. So on the left, we can see brown cells as GFAP positive cells for the never ventilate group, the middle for the mechanical ventilate group, and on the right, we can see a dot plot showing that we observe just above 10% for the never ventilate group, and 25, just above 25 for the mechanical ventilate group. So we conclude that there is neuroinflammation on the mechanical ventilate group with significance when we compare the group. The next marker that we used to observe neuroinflammation was IBA1 to show microglia. And why we use IBA1? Because in clinical and preclinical studies, it is correlated with acute cognitive impairment. So on the top left, we can see a slide for the never ventilate subject. On the bottom left, we can observe a slide for the mechanical ventilate subject. And again, brown means IBA1 positive cells. And on the left, we can see a dot plot showing the data for the never ventilate group and for the mechanical ventilate group. There, with a very considerable difference between the group, where we observe 10% for the never ventilate and 36 for the mechanical ventilate group. So again, our data, it is showing to us that the mechanical ventilate significantly more hippocampus inflammation with the never ventilate group. And the other marker that we analyzed, this is a new one. We analyzed GFAP in the serum. So serum concentration for GFAP. Remember that I showed to you that when we use GFAP in the tissue, in the hippocampus, we are analyzing neuroinflammation. But when we are measuring GFAP serum concentration, which means that we have to take blood and analyze this protein, that means that this cell has to die in order to release this protein. So in this case, GFAP in the serum is a marker for brain injury. In fact, FDA in 2019 just approved the use of this marker along with other markers, for example, to indicate CT scan for traumatic brain injury patients. We can see here showing the data for the never ventilate group, where we can observe 0.15, and data for the mechanical ventilate group, where we can observe 0.40. But people can maybe ask me, okay, Thiago, but what that's supposed to mean? Well, first, we are observing more brain injury in the mechanical ventilate group. But if we correlate this with human data, even though there are different species, the cutoff for a CT scan in humans, it is 0.15. So think about again, we taking a healthy young pig, ventilating for 50 hours, just a little bit more than 2 days, using the gold standard in ICU and mechanical ventilation. we are able to see brain injury. And the cutoff to intracranial hemorrhage, which means stroke. Hemorrhagic stroke is when the vessel, there's a rupture in the vessel inside the brain, and a lot of blood it is thrown inside the brain tissue. So this is a very catastrophic disease. So the cutoff for identify intracranial hemorrhage, it is 0.29. Of course, again, there are different species, but I'm using this data just to point out how mechanical ventilation can or might induce brain injury. So it is important to show again that our experimental conditions have not changed the gas exchange. So in order to show that, we use a ratio called PF ratio. So the ratio that we are oxygen and how this oxygen into the blood. So we measured this ratio. So the ratio was under the normal levels. And also when we analyze the lungs, we are able to show that the lungs are not damaged, which means that our experimental conditions have not induced lung injury, which would be a major confounder in our results. So the take-home message is the mechanical ventilated group had more hippocampus injury in our pig model, and where we observe higher hippocampus apoptosis. And in order to do that, we saw an increased hippocampus apoptotic index. Also, we observe higher hippocampus inflammation. And in order to observe that, we saw greater levels of GFAP positive cells and IBA1 positive cells. And also great brain injury, because we observe great concentrations of GFAP. I think that's it for me now.